This is Strides to Solutions. I am your host, Esther Adams. There is a small group of people studied now across several major research programs who reach their 80s and 90s with memory performance that matches adults 20 to 30 years younger. Researchers call them superagers, and the existence of this group raises an obvious question. If the typical aging brain loses ground in memory year after year, what is different about the brain of someone who simply does not? The tempting answer is that these are just people whose brains never really aged, that something protected them early and their neural tissue simply stayed young. That answer turns out to be only partly true, and the place where it breaks down is exactly where this gets interesting because it points toward a much more layered picture of what resilience in the aging brain actually looks like. Start with the structural evidence because it is genuinely strong and it is the most intuitive place to begin. When researchers scan the brains of superagers and compare them to typical older adults of the same age, they consistently find preserved gray matter in regions tied closely to memory and attention, particularly the medial temporal lobe, the anterior temporal cortex, and a structure called the anterior midcingulate cortex. In some of these regions, superagers are essentially indistinguishable from healthy adults decades younger. Cortical thickness that would ordinarily thin out with age instead holds steady. The hippocampus, a structure central to forming new memories, tends to retain more of its volume in superagers, and within the broader population of older adults generally, hippocampal volume tracks fairly closely with how well someone performs on memory tasks. White matter, the long fiber tracts that let different brain regions communicate, also looks healthier in superagers, with better microstructural integrity in frontal pathways specifically. Deep learning models that estimate a person's biological brain age from scan data confirm the same pattern from a different angle. These models consistently rate superagers as having a younger brain age than their actual chronological age would predict, and their rate of structural aging over time, tracked longitudinally, runs slower than typical peers. At the cellular level, the picture holds up under even closer inspection. Post-mortem studies of brain tissue from confirmed superagers, meaning people whose exceptional memory was documented before death and then examined afterward, find neurons in a memory-critical region called the entorhinal cortex that are significantly larger than those found in typical older adults, and in some comparisons even larger than those found in younger adults. These same brains tend to carry a lower burden of neurofibrillary tangles, one of the two hallmark protein abnormalities associated with Alzheimer's disease. Superagers also show a greater density of a distinctive class of neuron called the von Economo neuron, concentrated in the anterior cingulate cortex, and better preserved cholinergic signaling, a neurotransmitter system heavily involved in attention and learning. Taken together, this is a coherent, well-replicated structural and cellular story. Superagers appear to resist the tissue-level changes that erode memory in most aging brains, and the resistance shows up at every scale researchers have looked, from whole brain volume down to the size of individual neurons. So it would be reasonable at this point to conclude that superaging is essentially a story of a brain that simply did not age the way most brains do. But there is a finding buried inside this same body of research that complicates that conclusion in a genuinely striking way, and once you see it, it is hard to unsee. Superagers outperform typical older adults on episodic memory by a margin so large that researchers often describe it as equivalent to a 20 to 30-year gap. If you tested a group of 90-year-old superagers and a group of typical 65-year-olds side by side, the superagers would often perform just as well or better. That is an enormous functional difference. But when researchers calculated the actual structural brain age gap between superagers and typical older adults, using the same deep learning models that had shown superagers to be biologically younger, the difference came out to only about 1 year. 1 year of structural difference is being asked to explain a 30-year difference in memory performance, and those two numbers simply do not fit together cleanly. Something else has to be doing a substantial share of the work. This is the moment where the research shifts from a story about resistance to a story about compensation. And the distinction matters enough that it is worth being precise about the vocabulary researchers use, because these terms get used loosely in casual conversation, but they mean something specific in this literature. Brain maintenance refers to the relative absence of age related brain changes in the first place, essentially, the resistance story we just walked through, a brain that simply avoids accumulating damage. Brain reserve refers to baseline differences in brain structure, such as having a larger number of neurons or synapses to begin with, which gives a person more capacity to absorb damage before it starts affecting function, the way a wider margin gives you more room before you hit the edge. Cognitive reserve is different again. It refers to the brain's ability to actively work around pathology once it is already present, recruiting alternative neural networks, using more efficient processing strategies, or drawing on a lifetime of accumulated cognitive skill to keep performing well, even as the underlying tissue is not entirely intact. A useful way to hold these apart is that maintenance and reserve are both in different ways about the condition of the hardware, while cognitive reserve is about how skillfully the system can keep running software on hardware that is no longer pristine. The 1 year gap in structural brain age against a 30-year gap in memory performance is essentially forcing the field to take cognitive reserve and compensation seriously as a major independent contributor, not just a secondary factor layered on top of structural preservation. And there is direct functional evidence supporting this. When researchers put superagers and typical older adults through memory tasks while scanning their brains in real time, successful older performers show greater activation in the prefrontal cortex and hippocampus during encoding, along with a pattern of compensatory frontal recruitment that scales with how well someone actually performs, meaning the extra activation is not just noise, it appears to be doing real cognitive work. Higher cognitive reserve is also associated with slower cognitive decline, independent of how much brain volume a person actually has left, which means individuals with strong reserve can tolerate a smaller, more damaged brain while still maintaining a level of function that a person with less reserve could not sustain under the same structural conditions. Resting state EEG studies add another layer to this, finding that older adults with higher cognitive reserve preserve a property of brain networks called small world topology, a kind of efficient, well organized connectivity pattern that is usually associated with younger brains, suggesting that reserve is not just a metaphor for skill, but corresponds to something measurable about how the brain's networks are actually organized. It is worth pausing here to note that even the functional connectivity evidence is not entirely consistent, and a careful account of this research has to include that inconsistency rather than smooth it over. One well designed resting state imaging study specifically looking for differences in large scale network connectivity between superagers and typical older adults found no significant differences at all, which suggests that functional connectivity, at least as measured in that particular way, is not the primary engine of the superager phenotype either, so neither structural preservation alone nor functional connectivity alone fully accounts for the size of the memory advantage researchers keep documenting. That gap between what has been measured and what would be needed to fully explain the effect is one of the more honest and important open questions in this entire field, and it strongly suggests that superaging is not one single mechanism waiting to be pinned down, but the product of several partially independent systems working together, with the balance between them potentially differing from one person to the next. That idea that superaging may not be a single phenotype, but several different routes converging on the same outcome, has real support once you look across the broader evidence base. Some superagers appear to fit the classic structural preservation profile most strongly. Others show comparatively unremarkable brain structure, but score unusually well on markers of cognitive reserve and network efficiency. A separate line of evidence points to metabolic resilience as another distinct pathway, with some superagers showing higher concentrations of a molecule called N-acetyl aspartate in the posterior cingulate, a marker of neuronal metabolic health, alongside efficient glucose metabolism more broadly. Blood biomarker research has identified panels of markers spanning metabolism, hormone levels, and inflammation that can help distinguish superagers from typical peers using machine learning models, suggesting systemic, body wide physiology plays a role alongside whatever is happening locally in brain tissue. There is even preliminary genetic evidence pointing to a specific gene called MAP2K3, whose variants have been associated with superior memory performance through pathways related to how the brain clears or responds to amyloid protein, the substance that forms the plaques characteristic of Alzheimer's disease. None of these pathways appear to be necessary or sufficient on their own. Instead, the picture looks like a set of overlapping, partially independent routes to the same functional outcome, which is part of why studies searching for one universal superager signature keep returning inconsistent or partial answers. They may be looking for a single answer to a question that actually has several different correct answers, distributed unevenly across different individuals. Genetics also appears to interact with environment and lifestyle in ways that complicate any simple story. One well known genetic variant, a version of the APOE gene called the epsilon 4 allele, is associated with substantially higher risk of Alzheimer's disease. Interestingly, research on cognitive reserve has found that carrying this higher risk variant actually strengthens rather than weakens the relationship between certain lifestyle factors, such as mindfulness practice and social engagement, and measured cognitive reserve. In other words, the people who carry the highest genetic risk may, in some cases, get the largest measurable benefit from these particular protective behaviors, which is a useful reminder that genetic risk and modifiable behavior do not operate as two separate, independent tracks. They interact, sometimes in ways that run counter to intuition. There is also a broader finding worth noting on the survival side of this picture. Among people who live past roughly 95 years old, genetic influence on survival itself appears to grow stronger, suggesting that centenarians as a group likely carry some combination of protective genetic factors that support not just longevity, but resistance to the neurodegenerative processes that would otherwise erode memory along the way. Now, this naturally raises the question everyone actually wants answered, which is whether any of this is something a person can influence or whether it is simply a matter of the genetic and structural hand someone was dealt. This is where the evidence becomes genuinely more complicated than popular coverage of superaging often suggests, and it is worth walking through that complication carefully rather than skipping past it, because overstating certainty here would not be honest to the research. Physical activity is the modifiable factor with the strongest and most consistent association with cognitive resilience across this literature. Higher levels of physical activity are linked to more than double the odds of qualifying as a cognitive superager in some analyses. 6 months of moderate aerobic exercise produces measurable improvements in executive function, along with increased gray matter volume in the prefrontal cortex and temporal regions. Long-term studies following people for a decade find that physically inactive individuals show steeper declines in memory and executive function compared to consistently active peers, and this pattern holds even after accounting for other known cognitive risk factors. At the level of individual superagers, physical activity levels correlate with white matter integrity in a specific structure called the corpus callosum, which connects the two hemispheres of the brain, and one large analysis found that faster movement speed, not just self-reported exercise frequency, was one of the strongest phenotypic markers distinguishing superagers from typical peers. But here is where honesty about causal direction becomes essential, because the relationship between physical activity and cognition runs in both directions at once, and untangling which direction matters more is genuinely difficult using the kind of observational data most of this research relies on. Physical movement is a voluntary behavior. That single fact means cognitive decline itself can reduce how physically active someone becomes, since people with weaker executive function tend to have more difficulty with the planning, motivation, and follow through that sustained exercise requires. This creates the possibility of a cycle running in the opposite direction from the one usually assumed, where declining cognition leads to less activity, and less activity then contributes to further decline, rather than inactivity being the original cause. Randomized controlled trials, which are better equipped than observational studies to establish causal direction, do show real benefits of exercise on cognitive function, which is meaningful evidence that the relationship is not purely a matter of reverse causation. But the effect sizes in these trials tend to be modest, and some well designed longitudinal studies have found no significant association between exercise and later cognitive performance at all. The most careful summaries of this literature conclude that physical activity almost certainly plays some genuine causal role, but that its contribution is probably smaller and more entangled with other factors than popular narratives around exercise and brain health often suggest. Social engagement tells a similarly layered story. Strong social relationships and social participation are consistently associated with better cognitive outcomes over time, and some models even suggest social support carries more statistical weight than physical activity in predicting cognitive reserve. Social activity appears to influence cognition through multiple overlapping pathways at once, partly through direct engagement itself, and partly indirectly by encouraging more physical activity and supporting better mental health, which then independently support cognition. But the same reverse causation problem shows up here too. Cognitively sharper individuals may simply find it easier to sustain rich social lives, meaning the social engagement could be as much a consequence of good cognitive functioning as a cause of it. Depression is a further piece of this same puzzle, since depressive symptoms are associated with nearly double the odds of failing to reach superager status in some analyses. But depression itself can be both a cause of reduced engagement and a consequence of early, subtle cognitive changes a person may not yet be consciously aware of. Two large, well conducted cohort studies looking directly at the lifestyle habits of confirmed superagers actually reached contradictory conclusions. And I think that contradiction is one of the more important findings to sit with honestly, rather than resolve prematurely in favor of whichever result sounds more encouraging. A very large analysis using the UK Biobank dataset, examining thousands of superagers, found that they were actually somewhat less physically active than typical cognitively normal peers, more likely to smoke, drank more alcohol, and had worse adherence to a diet pattern generally considered protective for brain health. A separate, smaller cohort from the long running Whitehall 2 study found essentially the opposite pattern, with superagers engaging in more vigorous physical activity and reporting higher social engagement than their typical peers. Two carefully conducted studies, using the same basic definition of superaging, arrived at meaningfully different pictures of what superagers actually do in daily life. That is not a minor inconsistency to wave away. It suggests either that the lifestyle correlates of superaging differ across populations and study designs in ways not yet well understood, or that lifestyle factors, whatever role they play, are simply not doing the heavy lifting that popular explanations of superaging often assume. There is also a specific hypothesis in this literature worth naming directly because it has a certain intuitive appeal and turns out to be less supported than most people assume. This is the idea, often summarized as use it or lose it, that staying mentally active through activities like reading, puzzles, or continued intellectual engagement actively protects the brain against decline. when researchers have specifically tested for an interaction between age and mental activity on cognitive performance, meaning looking for evidence that mentally active people decline more slowly than less active people as they age. Very few studies actually find that interaction. An alternative explanation, sometimes called the preserved differentiation hypothesis, proposes something almost the reverse of the popular story. It suggests that people who stay more mentally active throughout later life were often already higher functioning cognitively earlier in life, and that their continued engagement is a reflection of that earlier advantage rather than a cause of later resilience. This does not mean mental engagement is worthless. Cognitive training studies do show real, measurable improvements, and interestingly, the pattern of who benefits most differs by cognitive domain. Training focused on episodic memory tends to produce larger gains in younger, higher functioning older adults, while training focused on working memory and executive function tends to benefit older adults starting from a lower baseline, which is itself an encouraging sign that the aging brain retains meaningful plasticity even quite late in life. But the specific, simple version of use it or lose it, framed as a straightforward preventive mechanism against decline, does not currently have strong direct empirical support. There is a deeper cellular mechanism underneath some of this resistance that is worth explaining on its own terms because it connects the structural story to something happening at the level of individual newly formed cells. The hippocampus continues producing new neurons throughout adult life, a process called adult neurogenesis, and research using animal models has found that cognitive resilience in aging tracks closely with whether these newly formed neurons survive over the long term and remain properly integrated into existing neural circuits. Resilient animals maintain healthy synaptic input into these long lived new neurons and preserve normal mitochondrial function within them, essentially keeping the cellular power supply intact. Animals that show more vulnerability to cognitive decline, by contrast, show reduced synaptic input and mitochondrial deficits in these same neurons, a pattern that researchers link back to dysregulation in the body's stress hormone system. This gives the structural preservation story a plausible cellular mechanism to point to, rather than leaving it as a purely descriptive finding about volume on a brain scan. Psychological factors add a further, somewhat unexpected layer to the resistance side of this picture. One study measuring what researchers call eudaimonic well being, a form of psychological well being tied to a sense of purpose and meaning rather than simple moment to moment pleasure, found that people with higher scores on this measure showed better cognitive functioning than their level of underlying brain pathology would predict, even after statistically accounting for known cognitive reserve factors like education. That finding suggests psychological well being may function as its own semi independent buffer against the effects of accumulating brain pathology, sitting alongside, rather than simply overlapping with, the more commonly discussed reserve factors like education and occupational complexity. One of the more revealing gaps in this literature is not a finding at all, but an absence. Despite the volume of research on structural preservation, on cognitive reserve, and on functional connectivity separately, remarkably few studies have directly connected functional network activity to the underlying cellular pathology within the same individuals. Reserve theories propose that people maintain cognition by flexibly recruiting alternative brain networks despite accumulating pathology, and structural studies separately document that some individuals resist that pathology altogether. But almost no research has combined detailed brain imaging, functional connectivity measures, and postmortem tissue analysis in the same people to actually trace how these systems interact across a single case. That gap exists largely because it requires an unusually demanding study design, following people with detailed imaging for years and then obtaining permission to examine their brain tissue after death, and few research programs have the infrastructure to do both well. Until that kind of combined evidence exists at scale, claims about exactly how compensation and structural resistance interact will remain somewhat provisional, built more on parallel bodies of evidence pointing in a consistent direction than on any single study that traces the full mechanism from cellular pathology through network function to preserved memory. Given all of this, it is worth stepping back and asking what a fair current summary of the evidence actually supports, because the honest answer sits somewhere between two extremes that are each easier to state than the real picture. It is not accurate to say superaging is simply genetic luck, a matter of some people winning a structural lottery at birth and everyone else being largely powerless to influence the outcome. The evidence for real, if modest, causal contributions from physical activity, social engagement, and general cardiovascular and metabolic health is too consistent across multiple independent lines of research to dismiss. But it is equally inaccurate to present superaging as a straightforward, mostly behavioral outcome achievable through sufficient exercise and sufficient social activity, because the structural and cellular evidence, along with the genetic evidence, along with the contradictory findings between major cohort studies, all point towards something considerably more layered than that. What the research actually supports is a picture of multiple, partially independent contributing systems. Structural brain preservation matters, but by itself accounts for only a fraction of the functional advantage superagers show. Cellular resilience, visible in things like neuron size and tangle burden, adds another layer that operates somewhat independently of overall brain volume. Cognitive reserve and compensatory network recruitment appear to do a substantial share of the remaining work, allowing some individuals to function well even when their underlying brain tissue is not perfectly preserved. Genetics shapes vulnerability and, in some cases, interacts with lifestyle factors in ways that amplify their apparent benefit for higher risk individuals. And modifiable lifestyle factors, chiefly physical activity, contribute something real, though the size of that contribution appears smaller and more entangled with reverse causation than popular framing usually allows. A related line of intervention research points toward another route by which behavior might influence brain function directly, even if the effect size remains modest. Structured mindfulness training in healthy older adults has been shown to improve measured cognition while also strengthening the functional connection between the hippocampus and a region called the posteromedial cortex, both of which are part of a broader network implicated in memory retrieval. This is a small but genuinely encouraging finding because it suggests that at least some of the functional connectivity differences associated with resilience are not fixed at some earlier point in life, but remain somewhat modifiable well into older age through a specific, trainable practice rather than through general lifestyle change alone. The dissociation between brain pathology and cognitive function shows up most starkly in research on centenarians, people who live past 100, where autopsy studies provide a level of direct evidence that scans alone cannot offer. Long-term follow-up of a large group of centenarians found essentially no measurable cognitive decline over more than a year and a half of observation, apart from a minor decrement in memory. When researchers then examined brain tissue from this same population after death, they found numerous cases where substantial neuropathology, the kind of tissue damage strongly associated with dementia in younger populations, coexisted with genuinely intact cognitive function right up until death. That combination, meaningful pathology alongside preserved function, is essentially cognitive reserve made visible at the most direct level available to researchers, and it reinforces the broader argument running through this entire body of work. The presence of damage in brain tissue and the presence of functional decline are related, but they are not the same thing, and the gap between them is exactly where resilience, reserve, and compensation do their work. It helps at this point to widen the lens slightly because superaging is really a striking, extreme case sitting inside a much broader research tradition on cognitive resilience and aging generally, And that broader tradition has spent considerable effort just sorting out its own vocabulary. Beyond the maintenance, reserve, and cognitive reserve distinctions already covered, researchers also distinguish between what might be called resistance, meaning a person avoids developing significant brain pathology in the first place despite carrying known risk factors, and resilience in a narrower sense, meaning a person develops the pathology but maintains function anyway through compensation. There is also a useful distinction between what researchers call preserved differentiation and differential preservation, two patterns that can look superficially similar in a single snapshot, but reflect very different underlying trajectories. Preserved differentiation describes someone who simply maintains whatever cognitive advantage they had at some earlier baseline, carrying it forward largely unchanged. Differential preservation describes someone who may have started at a similar point to their peers, but experiences meaningfully less decline over time, meaning their advantage emerges gradually rather than being present from the start. Distinguishing these two patterns requires genuinely longitudinal data tracking the same people over many years, and it matters because it changes what question is even being asked. One is a story about an early advantage that persists. The other is a story about a later divergence that only becomes visible with time. Education and occupational complexity remain among the most consistently documented contributors to cognitive reserve, though even here the mechanism is more specific than it might initially seem. Careful analysis suggests education exerts its influence on late life cognitive function mainly by building a higher level of cognitive skill earlier in adulthood, a baseline that then persists into old age, rather than by directly slowing the rate of later decline. In other words, education appears to raise the starting point more than it changes the slope, which is a meaningfully different claim than saying continued intellectual activity in old age actively protects against future decline, and it connects back to the preserved differentiation pattern described a moment ago. There is also evidence that the way reserve accumulates differs somewhat between men and women, shaped by differing patterns of employment and family responsibility across the life course, which is a reminder that the pathways feeding into cognitive reserve are embedded in broader social and economic circumstances, not solely in individual behavior or biology. General physical health status also appears to function as its own semi independent contributor to brain maintenance, separate from the specific behavioral factors already discussed. Vascular and metabolic health, in particular, seem to operate along a somewhat distinct pathway, with conditions like diabetes showing a moderate but measurable association with reduced brain maintenance over time. And the relevance of movement extends beyond deliberate exercise routines. One line of research on what are sometimes called super movers, older adults with unusually fast and efficient gait, found that this group showed better late life cognition despite no measurable difference in underlying dementia related brain pathology compared to typical peers, suggesting that whatever protects cognition in this group may be operating through a general marker of physiological vigor rather than through brain pathology specifically. There is one more piece worth naming, mostly because it complicates the picture in an interesting way rather than resolving anything. Personality itself shows measurable associations with the superager phenotype, with some studies finding higher extroversion scores among superagers compared to typical older adults, alongside greater endorsement of positive social relationships. Whether personality traits like extroversion are a cause of the richer social engagement associated with resilience, a consequence of maintaining sharper cognition further into old age, or simply a marker of some underlying biological factor influencing both, remains genuinely unresolved, and it is one more example of how difficult it is to cleanly separate cause from consequence anywhere in this literature. What I find most valuable about sitting with the full complexity of this evidence, rather than reaching for the simplest available headline, is that it changes what a person can honestly take away from it. It would be easy to end an episode like this with a tidy prescription: exercise more, stay socially engaged, keep your mind active, and technically none of that advice would be wrong. But stated without the surrounding uncertainty, it would overstate how much control any single behavior actually grants, and it would understate just how much of the superager phenotype appears to be built from structural, cellular, and genetic factors that a person does not get to choose. The research does not support either fatalism or a simple formula. It supports something more honest and, I think, ultimately more useful, which is a recognition that resilience in the aging brain is assembled from several different, partly independent ingredients, some of which are inherited, some of which reflect a lifetime of accumulated brain structure and function that current behavior can only modestly influence going forward, and a smaller but genuine slice of which does appear to respond to how a person lives, particularly through physical activity and social connection, even if science has not yet fully worked out how large that slice really is, or why it seems to look different from one large population to the next. This is Strides to Solutions podcast. Thank you for listening.